bit too bright this side. Yeah, this one. All right. Did everybody watch this movie? Yeah. Which one is this? Ice Age. Ice Age. I think it's the first one, right? Most likely that the guy goes like the last melon, and then all of those dodo birds try to get the melon. You know which one? All right, so that's the last lecture. It's very packed. No white clicker today. Sorry. So we left here last time talking about cytochrome C, electron transfer, right? Electron carrier. So here, remember we mentioned some of those heme-containing cytochromes, right? So cytochromes are names of proteins that have heme group, right? Cytochrome C is one of them, and is very, very important for electron transport. Where is it? The cytochrome C is going to be important. It's going to be between electron transport, electron complex 3 and 4. Okay. When you look at a heme protein, so heme is a protein, has amino acids, right? When you look at a heme protein, you're going to see that in the center you have an iron molecule in case of cytochrome C, okay? And that iron is extremely important to transfer electrons. So how does it do? When it is in the oxidized form, it is going to have 3 positive charges, so that's the ferric state. When it is in a reduced form, it's going to have 2 positive charges. That's your ferrous state. Oh, yeah, I know. No iClicker today, sorry. But I will give you, the sample questions and work through the answer. So this question is saying, oh, and the bonus is that I'm going to ask something very, very similar to this in exam number 4. Okay? All right, so here, which of the following is an electron carrier in photosystems that has a heme group? So here you have options for ubiquinone, cytochrome C, hemoglobin, myoglobin. So 3 of those have heme groups. One of them doesn't. That one, ubiquinone, is a very important electron cup, right? Remember you have the dione or the diol, selene-dione, dione, selene-diol with the OH, right? Alcohols. So it's not ubiquinone. So here you're left with 3 answers that have heme groups, and of course the one that is important for photosystems is your cytochrome C. Another thing about cytochrome C, it is water soluble. It is not lipid soluble. So as a water soluble thing, it is going to be floating outside of the membrane, right? And also, this is not only important for electron transfer between 3 and 4, it is also a signaling molecule for cellular stress. So when your cell comes to the end of its life, one of the ways that it can commit suicide is by breaking up the outer membrane of mitochondria and by liberating cytochrome C out in the cytoplasm. Okay? That is a way that cells can undergo apoptosis, which is programmed cell death, and is also a non-inflammatory type of cell death. So it's cell death that does not cause inflammation. I'm mentioning that here because, of course, we're not going to have time to cover apoptosis in this class. Okay, so here is the overall picture for the electron flow, right? So we know that between complex 1 and complex 2 and 3, you're going to have here your CoQ enzyme, right? So here, this is the one that is lipid soluble. So it's going to be inside of that membrane leaflet, okay? So that guy is your electron cup, and is going to transfer electrons from complex 1 2, complex 2, complex 3, and also complex 4 that is not showing here. So CoQ, coenzyme Q, can transfer electrons from complex 1 to any of the other complexes. Okay? After complex 4, you're going to have the transfer of electrons to the final electron acceptor, which is oxygen. When you transfer that to oxygen, then you're going to make water. Okay, this is a more reduced form. Okay. So here is that whole flow of those electrons. Okay. So remember here you have your CoQ that is derived from cholesterol. Okay, and that's your lipid-soluble one. And remember that I also had mentioned that you have certain supplements that can give a bit of a power up to CoQ function. And that is your CoQ10. Okay? And this is the one that you can get in health food stores or these online suppliers for health supplements. And it's interesting because, check this out, okay? CoQ is derived from cholesterol. So your cells have all this metabolic pathway that can make up CoQ for proper electron transport chain function. Now, have you heard of drugs that inhibit cellular production of cholesterol? Statins, yes. So you have atorvastatin, simvastatin, lovastatin, all those statins. What those drugs do, they are going to lower your anabolic process of pathways that make cholesterol inside of your cell. But remember that cholesterol can come up from 3 different sources, right? You can eat stuff that has cholesterol, you can produce it in the cells, and you also have a storage of cholesterol in the cells. Now, when you're making coenzyme Q, you are relying on your cellular levels of cholesterol, mainly part of that cholesterol pathway. Now, check this out. When you take statins, you inhibit that pathway. That means, most likely, your levels of CoQ are also inhibited. That means the electron transport chain is going to be deficient. So doctors should, and practice nurses, right? They should always advise the patient when they are taking a statin drug to also take CoQ10 for supplement to help to boost the formation of CoQ and the function of CoQ. Okay. Another thing about statins, I don't like them, especially for women. Basically, the literature says that it's very, very close to useless, if not totally useless. For men, there is a very low percentage of benefits, but I don't like them at all. I think very soon they're really going to go down in the popularity. Okay. All right, so things that are important to know here. Your CoQ is your electron cop. It transfers one by one to the other systems. It can receive electrons from complex 1 as well as from complex 2. It will transfer to complex 3, complex 3 and complex 4. Okay. Now, from 3 and 4, this is where you are going to have here your cytochrome C. And look, this is on the outside part of the membrane. It's not on the inside part of the membrane. It will associate to the membrane as a peripheral protein, but it's not going to go inside of the membrane. Okay, it's peripheral. It's on the outside. And that's the one that you never find in any supplement place. You're never going to find cytochrome C supplementation because that could kill some of your cells, telling them to undergo apoptosis. Bad idea. All right, so now we get to the fun part here on how to make ATP. So this is your complex 5, okay? It's also called ATP synthase. All right. So here, look at what's happening here. You have your membrane. This part here is your intermembrane space. So up here you're going to have the membrane, okay, outside the mitochondria. And this part here, they got charged with these protons. Right? So you have this proton motive force. You have a high concentration of protons here, and they have to go somewhere. So when they go through this complex, they're going to do something. The base of this complex is your F0. That thing, that part is the part that moves. Okay, I always get a bit confused there. Okay, your F0 part is the part that moves around. It is anchored by this other complex here that's called B2, but I don't care whatever it's called. It's an anchoring complex. It's anchored to one part here that doesn't move. Okay, so this F1 head here that says alpha and beta subunits, that part does not move. This one here is the part that moves. Okay, connected to this, there is also this stalk that is barely visible over here. So there is a little stalk over there. This stalk moves with the F0 place thing. Okay. Now this, when you put a proton through this, it will move this engine like a turbine. Okay. Also, you can imagine if you have a river and you have those water flow systems that generate electricity. You know what I'm talking about? Water mills, something like that. So this is like a water flow that it goes through the mill and turns this little engine. Okay, so it will go from the mitochondrial intermembrane space and it's going to go towards the matrix. Okay, so this part turns, that part turns, this part does not turn. But then this is the part that is going to make ATP. Okay, so here, this is just looking at it upside down or right side up. This here is the intermembrane space with the proton gradient, and here is your matrix. Okay, so these are the subunits of the F1 part. And let me see what is going to happen here. I just mentioned that. Okay, so this is the part here that is also interesting. With your electron flow, after complex 4, after you have pumped your protons into the intermembrane space, you have to get rid of those electrons. And at this point, the electrons do not really have a lot of energy, okay? But something has to pick them up. Your ultimate electron acceptor is going to be oxygen, and you're going to reduce the oxygen to make water. Remember, oxygen is very electronegative, so you need something that is very powerful to suck up those electrons to make water. Okay, so after complex 4 is when you have the formation of water. Now, when you have here your proton motive force, or you have a proton gradient, and it goes through complex 5, the process that you use to make ATP is called oxidative phosphorylation. Actually, it's called here chemiosmotic process. So you are working with a change in gradients in order to synthesize a molecule. So that is called chemiosmotic process. And overall, the synthesis of the ATP is done at a substrate level. Okay, you call that oxidative phosphorylation. Okay, this is to distinguish from substrate level phosphorylation that you have seen in glycolysis. Okay, so when you make this ATP here, when you put a phosphate to ATP and you get ATP, this is called oxidative phosphorylation. Okay, so different ways of making ATP, and of course, this process depends on oxygen, so this is your aerobic process to make ATP. All right, so here, interesting thing about your complex 5. So this is that F1, let me see. I think that it's, yeah. All right, what is the thing that I want to mention here? Remember this stalk that is connected to F0, that stalk moves. So when the stock is moving, this is a stock right here, okay? When the stock is moving clockwise, it is going to have an impact on those subunits that are part of your F1 complex, okay? So when this thing goes this way, when it hits the beta subunit, it is going to crank up this enzyme to allow for synthesis of ATP. So it goes clockwise, it's going to get ADP and phosphate, and it's going to make ATP. But this thing here, this subunit, beta, is an enzyme. And the conformation of the enzyme is going to dictate whether you put a phosphate to ADP or whether you push the reaction to go backwards. So this thing, in theory here, it could take ATP and break it down into ADP and phosphate. So what determines whether this enzyme is making ATP or hydrolyzing ATP is really the direction of that stalk and how it moves in relation to those subunits of F1. Okay? So going clockwise, you are synthesizing ATP, but if your proton gradient is shifted and now you have more protons in the matrix instead of the intermembrane space, then the flow of the protons can go backwards, and then this thing could turn the other way, which means you would be hydrolyzing ATP instead of making it. Okay, so this is all this very, very clever nano machine that is based on physical properties in order to allow this enzymatic reaction to take place. So here's the other question. How does your cell decide whether you have to make ATP or whether you have to put this ETC business to rest for some time because you have plenty of ATP in the cell? Well, basically, you're going to look at the ratio of ATP to ADP. Okay? Because remember, you're always recycling that nucleotide. Okay? So if you have ADP, you're going to put a phosphate and recover your ATP. Right? So if you have a high concentration of ADP in your cells, that means you're really using up ATP. When would be a situation that, that this would be the case? When you are exercising, right? So when your muscles are really pumping those ATP into your myosin heads and you are really burning up that energy, you have a lot of ADP that is formed. So that would tell your cells that they need to synthesize more ATP by oxidative processes. Okay, so when this ratio here, it tells you that you have more ADP than you have of ATP, overall your body is going to sense that and is going to cause you to breathe faster. Your respiration rate is going to increase. At the same time, you are going to allow more entrance of ADP into a mitochondria, and that is going to accelerate the production of ATP for recycling. Okay? So here you have what happens in case you have a resting state in comparison to exercise state. But how do you get the ATP into the mitochondria? And then how you get the ATP out of the matrix, right? So you have to have a system that is highly effective. So here it is not showing, but you do have this ATP and ADP anti-port system. Okay, so this is a process that does not depend on usage of ATP. But let's put a channel over here. Okay, so this is a channel, and here is that kind of same channel that is in the inside and the outer membrane. So here you're going to exchange this, and then you're going to get your ATP right like that. So ADP is going to go inside to reach your mitochondria, and then ATP is going to be delivered to the outside of that mitochondria, okay? And of course, this thing is an anti-port system. You have here your first stage, that is glycolysis, and that is anaerobic. Then you have your second stage, that is pyruvate oxidation, right? And also that occurs in the matrix. Remember how you get pyruvate to the matrix, right? So this is your second stage, this is your first stage. And then you have your third stage, that is the Krebs cycle. And then you have your fourth stage, that is electron transport chain. Remember what you're producing in every one of these stages. So with glycolysis, you're going to produce an ADH, and you're going to produce ATP, right? Not a lot of it. And then here, for pyruvate oxidation, you're going to produce some NADH. And then when you get here to the Krebs cycle, you're going to produce an ADH, FADH2, and you're going to produce energy that is ATP equivalent, but you're really producing GTP, right? Remember that here, that is like, okay, it's GTP, but is equivalent to energy of ATP, right? And then here, when you go to the electron transport chain, this is where you're going to recover your NAD plus and your FAD, to go back and pick up more electrons, protons, right? And this is where you're going to get most of your ATP produced. So here's another simple question. What drives the chemiosmotic generation of ATP? And the answer should be very obvious here, which is the difference in proton concentration. And as I joked with my last year class, I took this question out of ChatGPT. And if you have been using ChatGPT for practice questions, you notice that if you don't give it the proper prompt, it will always give you the correct answer as the longest one possible. Yeah, it's so annoying. So you gotta give it a proper prompt. Okay. So what is driving this? This is the concentration gradient of protons. All right, so here we get to the part that is my least liked part on photosynthesis. I don't like it, but it's necessary. All right, so let's look at this in a very summarized and easily digested, like pun intended, type of slides, okay? So you have here an interplay of what happens with cellular respiration, and then you're going to have to get your substrates that are going to come from this other process that is your photosynthesis, right? So here you're going to produce the products that are going to be used for mitochondria. Mitochondria is going to produce the products that are going to be used by chloroplasts in order to build up bigger molecules that are carbon-based, and those molecules can be used for production of energy. All right. And we knew that before. So now we're going to look at the really important things about those photosynthetic things, right? So looking overall at all the species that are living in the earth, like on the earth, right? We have most of the species that are producers. They are considered as producers. So they are going to use mainly light energy to build up some molecules that us, the consumers, can use for energy, okay? So those producers, they are not only building up these more complicated molecules based on carbons, so they're not just building up the anabolic process, but they also use mitochondria for their own cellular energy demands. Okay, so they're also using ATP that is produced by mitochondria, okay? So these guys here, they make the energy and they use up energy as well. We just use up energy. Okay. So similarities between photosynthesis and respiration process. Okay, you are going to be shuffling a chemical energy. Okay. When you look at the ATP that is produced because of photosynthesis, you call that process Look at this, phosphorylation. It's a lot of F's, and this is why I say it's good that you don't sit too close to it, but phosphorylation, that means speeding stuff up. Okay, phosphorylation. Use photons to produce ATP. Okay? When you look at the stuff that goes on in mitochondria, you call that oxidative phosphorylation, because you're going to use oxygen to accept your final electrons in order to produce the ATP. So here is when I tell you, clue is going to be in the test. Okay. So everybody's awake. Have you had your coffee today? Some people don't drink coffee. I don't know how this works. Okay. So, all right. So similarities between chloroplasts and mitochondria. Okay. You are going to have electron carriers, okay, and you're going to have several membranes. Like you look at the mitochondria, you're going to have two membranes, double membrane system. You look at your chloroplast, you're going to have three membranes. You're going to have those two membranes on the outside, and you're going to have a third membrane that is going to make those thylakoids here. Okay. When you put a lot of thylakoids together, you get this granule. Okay. Okay. Now let's see here. In both of those systems, you are going to use electron transport chain to make ATP. Okay. You make ATP in the chloroplast as well. Okay. Because the chloroplast and the cell that the chloroplast is inside of, these cells also need energy, okay? But the energy that is made inside of the chloroplast is only used inside of the chloroplast. It's not used for overall cellular demands, cellular energy demands of the cell. For the rest of the cell, you also have mitochondria hanging around there, okay? So the plants or those producers, organisms, they are going to have chloroplasts and mitochondria that are going to produce oxidative phosphorylation for their usage of energy. All right. So your similarities, you have here your double membrane, right? Inside of the inner membrane space in your mitochondria, this is where you're going to have those complexes for chemiosmotic phosphorylation. And chemiosmotic is the same when you compare mitochondria and chloroplast. Here, those complexes are going to be inside of those thylakoid membranes, right? Still process of chemiosmotic phosphorylation. The gradient of protons in the mitochondria is in the intermembrane space. The gradient of protons in chloroplasts is going to be inside of those thylakoid spaces. So this is called the lumen of the thylakoid. Okay? This is where you're going to have a lot of those protons. And they're going to flow out of there in order to make the ATP. All right. So those chloroplasts, they are kind of cousins of the mitochondria. And they also have those circular DNA, right? So chloroplast is going to have the circular DNA here, and that matrix in comparison to the mitochondria that's going to have inside, or actually it's called a matrix, right? So this space here between the space of the thylakoids and the second membrane, this is where you're going to have your DNA. So here it's called the stroma, okay? Stroma, oh, it's very red. The stroma of your chloroplasts. Okay, so know where the pieces are. Also in the stroma, you're going to find the enzymes that are important for the Calvin cycle. It's also called the dark reaction cycle. No, it's just Calvin cycle. This is why I don't like photosynthesis. Gets me confused. All right. Okay, so what is happening here? Photosynthesis is going to happen in two stages. Okay, so here you have your light reactions, and there are two of them. And then you're going to have your dark reactions. Yeah, That is also called the Calvin cycle. Okay, so light reactions and dark reactions. This is what's going to happen in your photosynthetic process. Out of your light reactions, you're going to have a little bit of ATP generated. That is going to be used only for the chloroplast. And mainly, you're going to have NADPH molecules that get generated. Okay, the NADPH is going to be shuttled to the Calvin cycle reactions in order to use the high energy that is being transported by the electron carrier, right? This is the electron carrier. It's going to be used to build up sugar, okay? Now, it's interesting, look at this. This electron carrier is NADPH, is not NADH. So here's another important fact for you. When you're talking about NADH, like NAD plus electron carrier, that is mainly used for catabolic processes, breaking things down. When you're talking about NADPH, that is mainly used for anabolic processes, so building things up. In the body, in our bodies, we also use NADPH, and that's basically when we are going to build things up, like the pentose phosphate pathway. Does it ring a bell? Anybody took biochem? The pentose phosphate pathway, another one to speak loud. No? Okay, anyway, that thing uses an NADPH. All right. So here, the processes of photosynthesis. First, you're going to harvest the energy because you're going to use light. And then you're going to shuttle it to the dark reactions, and you're going to build carbohydrates out of that and everything else that the cell needs. Okay. Now, important things to know: the light reactions, those depend on light. Surprise. The dark reactions do not depend on the dark, and they do not depend on the light. Okay. When you call dark reactions, it means just that they do not need light in order for them to take place. So even when the plant is in the dark, those reactions are still going on. So those are the anabolic processes that will build up carbon molecules, polymers. So here your dark reactions here is actually called carbon reactions in the book, right? It is going to need ATP, is going to need the reduced form of that electron carrier in order to build up those polymers of carbon. Okay. So here's where the pain starts. Okay, get ready. Perception of pain can be different, right, depending on how you interpret it. Let's make it not painful. Right. First thing you're going to do, you're going to harvest energy from the sun, right? First system that you're going to hit in your light reactions is going to be photosystem 2. It is called photosystem 2 because it was discovered after photosystem 1 was discovered, and nobody decided to ever change their names. So photosystem 2 is going to come into action first, right? It is going to use a certain wavelength that is coming from the sun. Their wavelength is around 680 nanometers. And it is going to split water in order to steal electrons from it, okay? So you're going to get electrons from the water. Let's see stage by stage what is important to know. Okay. So look at this. Your 680 is right on the red scale for your visible light. Okay. So here you have a spectrum of visible light that is go from 400 to 700. So here you have your 680. Okay. The other photosystem is going to use light that is on this range over here, that is in the blue range. Now notice that those systems are not using anything that is here in between. Mainly green light and anything close to green light is not used. So the light that is not used by those systems, it gets dumped out, meaning it will push it out of their system because they don't need it. And that's the reason why plants look green. They don't need the green light, okay? But they need red, they need blue light. Now what is going to happen? Light energy is going to hit this atom in the middle of this heme-looking structure, okay? Now the atom here is not iron, it is manganese, okay? So light is going to hit that, it's going to excite that thing. Manganese is going to steal electrons from water and is going to change the conformation of this hydrophobic tail, whatever that is, and is going to break water and steal electrons, okay? And it's going to push to the rest of the system. Okay. So here is a picture showing the comparison of different wavelengths, right, that we have all over. We only see this little fraction here. Right? Now, if you go to larger wavelengths, that means you're going to have lower energy when larger wavelengths, right? So if you look at big wavelengths like radio waves, those are very low energy. And then it start getting higher and higher type of energy. Now infrared here is only showing like heat energy, but this is a bit misleading because everything emits infrared light. Your bodies emit infrared light, okay? So infrared here is a bit misleading. We put a radiator there, but everything emits infrared light, which is low energy. Then you get your visible, right? And then from this, the shorter wavelengths, the energy gets higher and higher. Okay, so then you go ultraviolet, X-ray, gamma rays, and those really go through your system, and they can cause, you know, a lot of issues if you're exposed to them for a longer period of time. All right, so now let's look at those electron transfers and what's going on. So here is a molecule that is showing that heme structure with the manganese, right? And it's going to get those, it's going to get the electrons excited by light. Let's get to the part that is important. Okay. Okay, so here, when you look at the whole system that is light dependent, you're going to have basically two parts for the light dependent stuff. You're going to have one part that is going to capture the energy from light. So those are going to be your antennas. And then you're going to be the part that is going to shuffle the electrons around. Okay, and that's going to be your reaction center. Okay. So here they are, this is from your book. You have the first part here is showing you two places, right, surrounding your reaction center. You're going to capture the energy of light, you're going to break water, and you're going to shuffle those electrons, high energy electrons around until those reach the reaction center. In the reaction center, you're going to have some special chlorophyll pairs that are going to manage those electrons. Okay? So here's a picture of your reaction center. You're going to have those special chlorophyll pairs that are going to shuffle the electron into some electron carrier. Okay? And this electron carrier is going to move around and deliver the electrons somewhere else. Right? So these I have just mentioned. All right, so here the first part of that photosynthesis, which is your photosystem 2. What is going to happen here? You have the manganese containing heme group, right? Heme chlorophyll here. It is going to get water and break it apart, and it's going to produce oxygen, molecular oxygen. Okay. Now it does this thing in two stages, right? So first, when it gets the first photon, is going to break up the water. It's going to have a radical of oxygen, and that is going to wait for the second electron, for the second photon to get in, to break another water and get the second oxygen, and then you're going to put it together in order to make molecular oxygen. Now remember, if you have free radicals of oxygen running around, those are highly reactive oxygen species, and they can cause a lot of trouble, right? So this system has to be very, very regulated. So here, things that are happening in your photosystem 2. This first guy here is your photosystem 2. So you're going to have sunlight hits that, chlorophyll, and you're going to get the electrons all excited, right? So you get electrons excited here. Actually, it's going that way. This is redox potential of the electron. So energy goes up this way over here. You get the electrons excited, the electrons are moving here from manganese. Manganese has to get the electrons back, so then it steals it from water. This is where you have your water being broken down, okay? So the electron goes up there, gets all excited, then it's going to be transferred to one electron carrier, and the electron carrier is inside of the membrane. Everything here is happening inside of the membrane, okay? Electron carrier here is your plastoquinone, okay? So plastoquinone gets the electrons and is going to shuttle this to one area in which is going to push hydrogens to a complex to make the proton gradient. It's not showing here exactly what is happening, but the complex here is called B6F complex. Okay, so you get electrons here, you get your proton gradient being formed. Then you are going to transfer that lower energy electron to another electron carrier that is going to take this into your photosystem 1. When the electron gets to photosystem 1, it is actually at a lower energy than is useful for it to do work. So at photosystem 1, you're going to use another photon of light to get the electron excited again. Okay, so when the electron gets excited again, it's going to go to a higher energy state. Then it's going to be transferred to another electron carrier that is going to reduce NADP into NADPH. Then this NADPH is going to go to your Calvin cycle. Okay, so what is happening here? First thing that happened, electrons get excited. Manganese containing heme breaks water. Electron gets really excited, goes to plastoquinone. It's not ubiquinone, but it's equivalent. Okay, plastoquinone. This guy here, plastoquinone, goes to B6 complex and creates proton gradient. Now, then it gets transferred to this other carrier, that's called plastocyanin, gets to photosystem 1. Photosystem 1 gets excited with light and pushes this to ferredoxin that finally transfers the electrons to make NADPH. Important things to know here also, besides your plastoquinone, is that this is what causes your proton gradient, right? And this is where you get NADPH. So here, for your photosystem 1, you're going to end up with NADPH. For your photosystem 2, you're going to end up with ATP. Okay? Everybody cool with that? I know I'm going quickly, but we need to. So here, the first antenna center that is going to capture light, which one is it? Photosystem 2. That guy. Okay. You see that dotted line over here? This is when you get electron cycling. Okay. So what is going to happen here is that when you don't have a lot of light available, you can get this photosystem 2, sorry, photosystem 1, photosystem 2 is over here. Photosystem 1 to keep on making ATP and keep on exciting electrons. Right, so this photosystem 1 over here can receive energy from the sun, excite the electron, and then instead of making an ADPH, this system can donate the electrons back to plastoquinone and to the cytochrome B6F complex in order to make ATP. Okay, so this guy is photosystem 1, right? So this thing over here, you can have this type of short-circuiting of the process in case you need some more ATP for the cell usage, for the chloroplast usage, and in case you do not need to be making more NADPH. So let's say that your Calvin cycle did not take care of all of the NADPH that was available, and you kind of have a shortage of this electron carrier that is reduced, that is oxidized. So in this case, you can shuffle back the electrons to keep on having some ATP synthesis and not really producing an ADPH. Right? This is called electron cycling. Yeah.
Uses plasma again.
Yeah. It does, because it has to deliver it to the system that is going to make the proton gradient. Okay? All right. So here, this is the slide that I was saying, to produce the ATP, the cyclic electron transfer, okay? You can go back from photosystem 1 to prostaquinone. Sorry, it's another molecule here, but okay, let's say prostaquinone delivers it to cytochrome B6F in order to get your proton gradient. Okay? There's a lot of information in photosynthesis. So you can go to B6F and get your proton gradient. Actually, the correct answer for the transporter is actually ferredoxin, but it's a transporter that goes to cytochrome B6F. I'm not going to ask you about that, but yeah, it's a transporter that goes back to B6F. So those are going to B6F. You're going to have your proton gradient that is going to go to an ATP synthase, and you're going to get ATP made up of that ATP synthase, and the ATP is going to be used only for chloroplast needs. Okay. This is what we just covered there, right? We just made that. Now we get here to the Calvin cycle. Important things about the Calvin cycle, okay? You're going to convert inorganic carbon dioxide, which is a gas, right? And you're going to make units of building blocks of carbohydrates, okay? Those building up reactions are going to be powered by ATP, and they're going to be powered by discharged electron carrier that is NADPH. The first enzyme of this pathway is the enzyme that is the most present, is most abundant in the world. That enzyme is called Rubisco. Okay, so this is the one thing I want you to remember from the Calvin cycle. Scary. Okay. So here you get carbon dioxide. Which form is that thing? State is gas. You're breathing it out. Breathe carbon dioxide. Right? It goes through the membranes readily. So this thing gets carbon dioxide. Okay? The enzyme that is going to be the first one that is going to synthesize stuff in this process, that enzyme here is the Rubisco that goes right there, putting carbon dioxide together here. Okay? You're going to get 3 molecules of a 5-carbon compound. You're going to make 6 molecules here of your carbon compound. Okay, then a lot of things are going to happen. I'm not going to torture you through this. You're going to use ATP, and you're going to use reducing power of an ADPH, and you're going to build up your carbon chains. Okay, what kind of carbon chains? It could be sugars, it could be carbohydrates, it could be fatty acids, it could be amino acids, anything that uses a carbon for building compounds. Okay? So what should you remember here? The first enzyme is your Rubisco. Okay? Where are things coming from? You get stuff from the light reaction. You're going to get some ATP, right? This is all happening in the chloroplast. You get ATP, you get NADPH, that is coming from the light reaction. So your light reaction has to supply those substrates for the Calvin cycle to take place. What is going to end up as a product of the Calvin cycle? You're going to get glyceraldehyde 3-phosphate, and you're going to get your oxidized forms of whatever substrate you used. So NADP and ADP. So here are your light reactions. So remember 2 of them. And here is your dark reactions. Here, just for fun, there was this photosynthesis is a divine type of song from a colleague, Kevin Ahern, that, you know, he's very, he uses a lot of imagination for those things. If you want to hear the song, go to the link and you know, distract yourself. So now, finally, we get to some points of the parts that I like the most. Yay. Okay, do we have major questions for photosynthesis before we go on? I know I'm rushing through it, but it's because I'm giving you the exam 4 on Friday, right? Yeah. First enzyme in the process is Rubisco. You use the products. Okay, the question was, what do you need to know about the Calvin cycle? Good question, right? What should you know? You're going to be using this for anabolic process. You're going to build up stuff. You need substrates that are coming from your light reaction. What do you need? You need ATP, and you need NADPH. That's your reduced form of that electron carrier. You're going to end up with glyceraldehyde 3-phosphate, high energy building block that is going to be used to make carbohydrates, lipids, and proteins, amino acids. That's all you should know about the Calvin cycle. Also know that it's also called the dark cycle, and you don't need light for it. It happens during the day, it happens at night, you just don't need the light for it. Okay? All right. Any other question about photosynthesis? Are we good? So 20 minutes of the part that I like more, right, which is cellular communication. Okay. So here, cells need to communicate. Everything that is alive is going to communicate. What is the smallest living unit? It's a cell. Are bacteria cells? Yes. Do bacteria communicate? Yes, they do. So all living forms communicate. Okay? So what is it that's going to use for communication? You can use some proteins that the cells are making. You can have cytokines. You can have stuff that you put out there, soluble factors. You can have cells that bump into each other and have receptors. Those are direct cell-to-cell communications. Those are very important for immune system functions. You can have big vesicles, you call them microvesicles. You can have tiny vesicles, you call them exosomes. And you can have a lot of speech bubbles in case you have bacteria. Bacteria communicate by speech bubbles. Okay? So here is a happy cell saying, you know, now we're talking. So here, how cells communicate. You have this cell on one side that gets the cell phone and say, hey, let's coordinate here on production of hormone. So the other cell for the same organ here is going to say, okay, well, how about in about 10 minutes? So they're all coordinated in a way that they're going to produce a certain protein or hormone or factor. Okay. So here is a link in case you're interested in bacterial communication, right? And I really like this this TED talk by Bonnie Dassler. Okay, so you can follow the link and see how bacteria is going to communicate. Okay, I'm not going to ask you about that, but this is stuff that I will ask you here. In different ways that a cell can communicate when they put stuff on the outside. So the first way they can communicate is auto-communication. So they can tell themselves something to do. This is applied when you're sitting, you know, just before exam, you're thinking, I think I should study. So you're telling yourself that you think you should study. That is a form of autocrine type of communication. In case of a cell, if it produces something and then it has a receptor for that molecule it produced, this is a type of an autocrine type of communication. Okay? Cell produces something and is exciting itself. That is autocrine communication. Now, the second stage of communication here is when a cell is producing something and is hitting the receptor of a cell that is close by. So this cell produces, let's say, a cytokine, and then you have other cells that have a receptor for that, and the cell captures the cytokine. So this close by, this proximity of cells is called paracrine communication. And then the third example here is when you have a cell like far away from a target cell that has the receptor for it. So in order for that communicating molecule, that is the first signal of communication, to leave the producing cell and get to the target cell, the way that it has to do is that it has to go into the blood circulation, and then it's going to go and hit the target cell. Okay, so hormones, a lot of hormones basically work that way. Okay, so this is an endocrine type of communication. When it gets into the bloodstream and hits the cell, that's endocrine type. So pathways that the cells can communicate. You have extracellular communication, right? So in this way, you're going to have a receptor that is on the surface of the cell. So here is going to be a receptor. And then a molecule gets there, and you communicate by that method, extracellularly. And then you have another communication that's called intracellular communication. Now you're going to have a cell. You're going to have your molecule that gets there, but the receptor now is inside of the cell. So this molecule has to go inside of the cell in order to hit the receptor. Okay. What kind of molecule is going to do that? Well, a molecule that is very small and hydrophobic, right? Like hormones, certain hormones can go through the membrane and hit the receptor that is inside of the cell. And then there are more flavors to that. Sometimes the receptor can be inside of the nucleus. So then your hydrophobic small molecule will have to go through all these other membranes to hit the receptor inside of the nucleus. So here's from your book, showing your surface bound receptor or your cellular bound receptor. So in this case here, you have a receptor that is inside of the nucleus. So intracellular receptor could be either cytoplasm or inside of the nucleus. And sometimes you can have a receptor that is in the cytoplasm and moves to the nucleus. All right, so here is the idea that all cells need to communicate. What kind of messages they send? They send some messages that allow cell survival. Other messages, they're going to tell the cell to divide. Other messages will tell the cell to modify its function. And then you have messages that tell the cell that it's time to die. So all this communication is important for function. So what kind of communication is sent and received? Sometimes a cell is producing this first messenger, and the first messenger will have different types of receptors depending on the cell that is receiving the message. Okay, so for example here of this cytokine that is interleukin-4, okay, if it goes to a receptor that is present on a B cell, then the B cell is going to interpret the message as something that is telling it to proliferate and to differentiate and to do something. Now, if this cytokine hits another type of immune system cell that is called a thymocyte, okay, or it's going to become a T cell, then this first messenger is going to tell the cell to divide. That's it, nothing else. And then when you look at another immune cell that is kind of mast cell, the receptor here is going to tell this cell to also proliferate, but it's a different type of cell, even a different type of receptor. Okay? So when you have one thing that is doing different things depending on the cell type, you call this type of signaling as pleiotropism. Okay? Pleiotropism, when one molecule can tell different cells different things to do. And then you have this case that different molecules can tell the same cell to do the same thing. So now you have here, for example, of interleukin 19 can go and tell this cell to get activated, or you can have interleukin 20 that can use the same receptor and tell the cell the same thing to get differentiated. So when different things do the same job in the same cell, you call that redundancy. Again, that's very important for immune system function. Okay? So redundancy of the effect. All right, sometimes this redundancy can have a more powerful effect on the cell that it hits. So you can have different cytokines that are doing the same job, but if you put them together, you get a more powerful type of signal. So you call that a synergistic type of effect. So it's bigger, it's better, it's synergistic. Okay. Some other times, you can have different signals, right? Different molecules, their first messenger, that are going to hit the same receptor, but instead of being additive, they actually antagonize each other. So in this case, you call it antagonistic effect. So whether this cell is going to be activated or blocked depends on which one of these signals is available. Usually, if you have both signals available, then your target cell is just confused. No, that's not true. Usually, if you have both signals, which one is going to predominate? Okay, let's take a guess. Is it the positive or the negative signal? Negative, who said that? Yes, negative, negative. If you have both signals available, the negative one is usually the one that wins. Okay? So antagonistic signals, if both are available, negative one is going to tell the cell not to do anything. Okay? Instead of the other one that is going to tell the cell to do something. Okay, so here's another example of a question. So growth factor released by a cell causes nearby cells. So here is nearby cells to grow. What kind of communication is that? Paracrine signaling. And here we get to those interesting different types of surface receptors. So we have seen this guy before, okay? Ion channel couple receptor. What was the example that we've seen this before? No. Ligand-gated channel. Like, you know, I'm telling my muscle to contract. Neuromuscular junction. Remember acetylcholine? It's the receptor, opens the sodium channel. It's not sodium potassium channel. Did you say just sodium channel or sodium potassium? I heard sodium potassium, but here's the sodium channel, right? When you have your neuromuscular junction, you put acetylcholine, you open the channel. We have seen that before. Now, this is the one that is interesting. G protein coupled receptors. You have your ligand that binds on the outside of this channel, changes the conformation of the inside of the channel that is coupled to these other proteins, then the other proteins are going to do something. Okay, so this is a part, G protein couple. And then you have this third part that is enzyme couple receptor. So you have receptors on the surface that are usually not dimerized or trimerized. They're usually monomers out there. But then your ligand molecule comes along and gets those two receptors to interact. When they interact, they have some type of enzymatic property in the cytoplasmic side. And this thing is going to cause something to happen to the cell. Okay, so those are enzyme-linked type of receptor. Okay. So here is just for your studying purposes. Okay. So here, on receptor subtypes, right? If you have something on the membrane, that is a cytoplasmic receptor, as I've seen before. If you have something that goes through the membrane and goes to the inside of the cell, those are going to be intracellular receptors. And here are two types. You can have cytosolic intracellular, or you can have nuclear intracellular. Okay. Let's get to the part that is important for you to know before everybody runs out. You can read that on your own. It's very self-explanatory. So what's going to happen? Your first messenger gets to your receptor. Your receptor is going to produce a second messenger, and this process of producing a second messenger, it is called signal transduction. Okay? After signal transduction, you're going to have your cellular response. Okay? Which molecule performs the primary signal transduction? That is your receptor. Okay, think about this question later. I want to get to the part that is important for you to know, which is here. G protein couple receptor. It is a transmembrane protein that is going to span the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. Okay? The extracellular part of this transmembrane protein has the receptor. Okay? So receptor is going to bind to it, is going to send a signal to the inside of the cell, and is going to tell some other proteins that are associated with the receptor, is going to tell those proteins to do something. So here is when you get your G protein. G protein coupled to the receptor. This is a G protein test. It's mainly made of 2 parts. Some books say 3, but it's mainly 2. You have your alpha domain, and then you have your beta gamma domain. Beta gamma are always together, so that's why I classify them as the second part. This thing is heterotrimeric, okay? So alpha, beta, gamma. So what is going to happen here? When your receptor binds to the ligand, you are going to turn this thing on. When you turn that thing on, the GTP binding area, which is always in the alpha subunit, is going to get GTP on it. How? Well, it can get on its own, or it can use some gas. You have heard the gas before, right? When it gets GTP, it is going to dissociate from the beta gamma. So this guy is activated, the alpha part. Beta gamma is also activated. They're going to do different things. Everybody so far? What is it that they're going to do? Well, it depends on the cell. You can have some G proteins that are stimulatory of some signal. You can have some G proteins that are inhibitory. And you can even have another flavor of a G protein, okay? So what you're going to do, you are going to produce second messengers that are going to tell the cell to do something. So when your second messenger is produced, you call that stage the amplification of your signal, okay? So here, this is a part that is important for you to know, so before you run out, right? So this is a type of receptor that is stimulating, and your G alpha is going to produce a second messenger that is called cyclic AMP. Okay? So cyclic AMP is going to be produced because of that because of that activation of the G protein. The enzyme that gets activated by the G alpha protein is an enzyme called adenylyl cyclase. Okay, so adenylyl cyclase produces cyclic AMP. Cyclic AMP does not hang around forever. It's going to be broken down by an enzyme that is called cyclic AMP phosphodiesterase. So here is the part that is assimilating, produces cyclic AMP. And you also have inhibitory signals that could just tell the cell to be quiet. I'm not going to get to that part, okay? How is it that cyclic AMP is going to work? Well, cyclic AMP is an example of your second messenger. Usually it is going to bind to some proteins that are inhibiting the action of another protein. So the example here is modulating the ability of protein kinase A to work. So when cyclic AMP binds to the repressor, then this enzyme can work, is not repressed anymore. So all this area is part of your signal amplification. Okay, I want to get to the favorite slide. You can read this on your own. Okay, this thing. Okay, I know I'm getting to my last minute, so hold on. If anybody has to run out of here right away, go ahead. I'm going to put in the video, right? So here, things are important to know here. When you produce your cyclic AMP, usually this is in a state that you do not have a lot of energy available. So cyclic AMP in certain cells in your body, especially in your liver, is going to activate the breakdown of glycogen in order to produce glucose, and then the glucose can be exported out of the liver cells and increase your blood glucose levels. Okay? So this is a way that you can get extra energy based on the glycogen that you have stored in your liver. Okay? Now, here, this is the part I like. How is this related to drinking coffee? So now let's say you are in a low energy state, you're producing cyclic AMP, and then you activate protein kinase A. That thing breaks down glycogen, you produce glucose. And then, if the system is just working on its own, you have that enzyme, phosphodiesterase, that is going to break your cyclic AMP into AMP, and then this signal transduction is going to stop. When you drink coffee, you are going to inhibit this enzyme. Now you make your pool of cyclic AMP available for a longer period of time. That means that after drinking a moderate amount of coffee, you're going to have more sugar present in your bloodstream, and you're going to have a bit of more energy left. Right? This was the last part that I wanted to mention here. Just make sure that that you follow the video that I'm going to put up. That is going to finish up with the rest of these slides. But basically, I'm not going to ask you anything past the coffee. All right?